So today I'll be talking about molecular and genomic approaches in the oral microbiome. So as we all know, the molecular and genomic approaches helps us in understanding how the oral microbiome actually works at the molecular level. So there are various tools and analysis that functional analysis that are being done to understand how this entire mechanism mechanism works. So they currently understand helps to understand how metabolic pathways and virulence pathways can be identified and studied, so that in future we can better create medicines and fight these oral diseases. So coming to the sequencing method, that is many advanced sequence technologies are used. So they have revolutionized how we understand the microbial pathways. So identification and analysis can be done using these sequencing methods. So the first one that we'll see about is the 16S RNA sequence, rRNA sequencing. So it primarily targets the 16S microbial RNA, ribosomal RNA gene. So that is highly specific among the bacteria. So each bacteria has its own kind of 16S rRNA. And it also is used for taxonomic classification because it is species specific, and it requires some bioinformatics pipelines such as QIIME and Mothur softwares, which helps to analyze and process the data of RNA sequencing. This method is actually cost effective, but it is widely used method for species taxonomy, but lacks resolution and at the strain level. So we can identify the species, but it is very difficult to identify the specific strain within the species. The next is shotgun metagenomics. This method involves sequencing of the entire microbial genome that is present to provide a more narrower species level identification by showing functional potential of the microbiome. This enables the detection of genes and associated with antibiotic resistance metabolic functions and the virulence factors. So these can be also mapped out in the genome sequence of the bacteria. This requires high computational power because because of the sequencing of the entire bacterial genome. So tools like MetaPHL and human megahit etc. are used for this. So this, due to being more computational, it is more expensive, and also it provides deeper understanding of the bacteria in comparison to 16S RNA sequencing. The whole genome sequencing, this focuses on understanding the entire sequence of the genome of the individual microbial species isolated from the oral cavity. So this provides a detailed genetic information that includes like structural variation, horizontal gene transfer and antibiotic resistance genes. So these can be analyzed by using this method. So this mainly helps in understanding the evolutionary history of the microbe and the genetic adaptations it has done over the period of time according to the environment. So after the sequencing and all, we come to what known as the metagenomics and microbiome data. So the data that has been collected from the sequencing has to be analyzed and interpreted for it to for understanding. It generates a very vast and big data and requires specific tools for understanding and interpretation. So the data analysis, the steps include quality control, trimming of low quality reads, and then removal of host DNA contamination. Then after this, taxonomic classification is done. So this is done using the reference of databases like Silva and Green Genes and Kraken. So taxonomy is naming the microbe. So the database already has the record and which has been uploaded on the software or the internet, which helps to identify the specific bacteria by matching the sample and the database. So data interpretation, the microbial diversity analysis to find how the how diverse the microbe is. So the alpha and beta diversity are the two metrics which is used to compare the microbes. And comparative metagenomics, it implies how identifying different microbial composition between healthy and diseased state. Then the microbial network analysis helps to study how microbe interactions occur and the patterns of co-occurrence of interaction to understand the ecosystem of the microbes. And some machine learning approaches are also there which can help to interpret data by using AI models. The functional metagenomics, it goes beyond taxonomy, which is a next step. It helps to identify the biological roles and microbial genes, namely for its metabolic and virulent pathways affecting the oral environment. So this helps to understand how the virulence actually occurs or the metabolic pathways of the microbes. So we'll study about the virulence pathways. Understanding this, it helps in identifying the gene that is involved in the pathogenicity, adhesion and invasion of the immune and immune evasion. That is how the microbe causes the disease, how does it adhere to the oral cavity, and how does it invade, and how does it skips from the body's immune system. So it also helps to understand various pathways such as oral infection, periodontal diseases, and cancer-related microbiome shifts. Detection of antibiotic resistant genes can be understood by using tools like Comprehensive Antibiotic Resistance Database, CARD, or RES Finder. The metabolic pathways, this allows to understand and investigate of the microbial communities and to understand how the metabolism of different substances occurs in the microbe, such as carbohydrate metabolism, amino acid, and also helps in assessing how things like how biofilm formation occurs. So this helps in identification of key enzymes involved in the amino acid production and pH regulation. It also involves in which also affects the oral health. Example, how in caries certain bacteria produces acidogenic byproducts that induce that decays the tooth structure. and causes caries. So that can be understood by understanding how the metabolism of the bacteria is. So databases are used such as Cake, MetaSig, and BioSig. So they have already stored the metabolic pathways which can be used for comparative evaluation. So the applications for bioinformatic tools in oral microbiome research. Firstly, it is the taxonomic profiling of profiling tools. So such tools such as QII, ME, Muther, and Metafel, and these tools helps in the identification of the species specific by comparing the sample with the database. The functional annotation tools such as human, MGRASH, and Megan. Then genomic assembly and annotation such as spades and proca. Statistical and machine learning tools such as R packages, Python based machine learning models, and for predictive analysis, and the database resources such as Silva, Green Genes, Cake, and IgM. So these all are the bioinformatic tools that can help in the microbiome research. So the conclusion here is that the molecular and genomic approaches are very important for understanding how the bacteria exist, and to understand how the virulence pathways and the metabolic pathways of the bacteria to better understand and give a proper treatment when in case of infections and diseases that occurs due to the oral microbiome. Thank you.